last time we decided to talk about two species, and then there were three types of interaction: predator-prey, competition, and mutualism. And we did predator-prey last time. So today we will do competition. And this is also a Lotka-Volterra model. But is now a competition model. Okay, so we have so we're going to have N1 and N2 as the species. So dN1 by dt in this model. So R1 N1 into 1 minus N1 over K. So that's our logistic growth model. Okay, and now the difference is that they're competing against each other. And so N2 decreases the growth of N1. And so that's represented by this term. So what we can see here is that, so the idea here is that N1 interacts with N2, and that's a negative effect, okay? And what does this B12 mean? Well, this is a competition of N with itself, and this is the competition of N2 with N1, and this gives you the sort of, how does that compare with this one, okay? And then same sort of thing, dN2 by dt equals R2 N2 to 1 minus N2 over K2. That would be just logistic growth. And then we've got a minus B21 N1 K2. Okay? That's the competition. And so what we do, so we could call this competition with N1. So call it sort of interspecies competition, or intra-species is competition with itself. And this would be interspecies competition. That with the other, with N2, and this one with itself. Okay, and now we do the usual thing. We non-dimensionalize, find steady states, draw the phase, do linear stability analysis, draw the phase planes, and that's it. Okay, so that's stuff that you should know to do from the DE's course, right? So I leave you to non-dimensionalize, and you end up getting D1. So that's going to be N1 U1 into 1 minus U1 minus alpha 1, 2 U2. We'll call that F1. And du by d tau equals rho U2, 1 minus U2 minus alpha 2, 1 U1, and that is F2 U1 U2. One thing I should have mentioned up here is that all the parameters R1, R2, K1, K2, B12, B21 are positive constants. Okay, so there were 6 parameters. We can do 3 scalings, n1, n2, and t, and so we reduce it to 3 parameters. Okay? Steady states. I leave you to do this, to check. U1 could be 0, and if U1 is 0, then U2 could be 0 or 1. Likewise, other way around, if U2 is 0, U1 could be 0 or 1. And the only nasty one is this one. It's where neither are 0, and therefore you've got these two linear equations, simultaneous linear equations to solve, and that gives you this, leave you to check this. Okay. And so that's going to be non-negative for the cases alpha 1, 2 less than 1 and alpha 2, 1 less than 1, or alpha 1, 2 bigger than 1 and alpha 2, 1 bigger than 1. And the way we get that is, well, if these are bigger than 1, that's negative, that's negative, that's negative, so it's all positive, so that's fine. Less than 1, or sorry, that got that the wrong way around. Less than 1 means that's positive, 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 that's fine. Bigger than 1, negative, 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 negatives cancel, that's fine. What happens if 1 is positive and 1 is negative? Then regardless of the sign of this, these will have opposite signs, which means 1 will be positive and 1 will be negative. And that's not realistic, okay? So, leave you to do the analysis, linear stability, and I'm just going to write down the answers and leave you to do the analysis. 0, 0 is unstable node. One, 0 is stable node if alpha 2, 1 is bigger than 1, and it's a saddle which is unstable if alpha 2, 1 is less than 1. Okay? Then 0, 1 is the same thing, but with the 1 and 2 switched round. Stable node if alpha 1, 2 is bigger than 1. Saddle if alpha 1, 2 is less than 1. Okay? So what this means is that if alpha 2, 1 is bigger than 1, which means that U2 or U1 has a detrimental effect on U2, big detrimental effect on U2, then U2 is driven to extinction and U1 wins. And likewise here, the other way around. Okay? And so the last one linear stability analysis is horrible. Okay, so I'm not going to do it. You can do it, it's pretty straightforward, but the algebra is horrendous. And instead, what we're going to do is go straight to drawing the phase planes. Okay? So let's come down here a bit. And now if we think about the phase planes, we've got U1 could be 0. Oops, got the wrong. So U1 equal to 0. That's U1 dot equal to 0. And U2 equal to 0 is for the other one. Okay? And if otherwise, we've got these two straight lines, 1 minus U1 minus alpha 1, 2, U2 equals 0, and likewise for U2. So these are lines that are going to have negative gradient. So if you think of two lines with negative gradient, then there's 4 possible cases. One is below the other one, and they don't intersect in the quadrant. So that's 2 cases, because one could be below, or they could switch around. But on the other hand, they could intersect. And there's 2 ways they could intersect. And therefore that gives you 4 cases. Okay? So here's the case 1, alpha 1, 2 is less than 1, alpha 2, 1 bigger than 1. So what do the trajectories look like? So if I've got this right, that should be U1 dot equals 0. So let's just check. That should be the line U2 equals 1 minus U1 over alpha 1, 2. So when U2 is 0, U1 is 1. When U1 is 0, U2 is 1 over alpha 1, 2, and that's bigger than 1. And then the other one be this. Oops, so that should be in blue. That should be in blue. One over alpha 2, 1, and 1. Okay. Let's see the steady states. Here's a steady state, 0, 0. Here's a steady state, 0. Here's steady state, that there. And remember, if they have different, less than 1, bigger than 1, the coexistent steady state doesn't exist. Okay? So if we start here, U1 dot and U2 dot are both positive, so we increase. We cross this line horizontally, because that's U2 dot equal to 0. And now U2 dot becomes negative. And therefore, we go here. Okay. if we started off here, both are negative. We cross this vertically, u2 is still negative, u2 dot is still negative, and we go there. Okay, let's just see if that's consistent with our linear stability analysis. 0, 1, if alpha 1, 2 is bigger than 1, 0, 1 is stable node. That looks stable node. And what about this one? Or sorry, this one here. 0, 0, 1 is a saddle if alpha 1, 2 is less than 1. Okay? And remember, a saddle has one direction that's stable, another direction that's unstable. And so what we would have is, for example, if u1 was 0, then we just have logistic growth for u2. So we would go like this or like this. But if we started here, we would go down like this. Okay? So can we understand this biologically? Alpha 2, 1 is big, alpha 1, 2 is small. Alpha 2, 1 is large, means u1 really goes for u2, really competes, makes life horrible for u2. Meanwhile, u2, alpha 1, 2 is less than 1, it's a bit kinder to u1. So u1 is a better competitor and u1 wins out. Okay? And this is called competitive exclusion. u1 is a better competitor. Okay, and really to understand this fully, of course, we should go back to our dimensional parameters, which is the same sort of thing. That's B1, 2. If you do the non-dimensionalization, you get that. Alpha 2, 1 is B2, 1, K1, K2. Okay. So if you didn't like U1 and you liked U2, then you would have to change these parameters in some way to move away from this case. Okay. Now we switch around. Case 2, which I leave you to do as an exercise. Alpha 1, 2 bigger than 1. Alpha 2, 1 less than 1. I leave that as an exercise. U1 is excluded. Okay, do these again. Now the red line is going to be below this one. So you'll go to U2 first. Okay. Or if you wanted to, you could just name U1, call it U2, U2, call it U1, and you're back to that case. Okay. 3. Alpha 1, 2 bigger than 1. Alpha 2, 1 bigger than 1. So U2, U1 equal to 0 is that one. And this one. Right, now alpha 1, 2 is bigger than 0, so this intersection is less than 1. And the other one, they're both bigger than 1, so there'll be there'll be a coexistent steady state. So there'll be this one, u2 dot equals 0, and this one, 1 over alpha 2, 1, and that's u1, u2 dot equals 0. And now we've got 4 steady states. Stable, or it's that one, always unstable, that one, and that one. And what's going on here, so they're both bigger than 1, which means these two are stable. And if you go through and do this horrible analysis, you will find out that this is the saddle point. So, suppose we start off here. We go up, because everything u dot 1, u2 dot positive. Then we cross that line, and now u2 dot becomes negative, and we go to there. On the other hand, if we start here, u1 and u2 dot both positive, we go up, but now we cross the red line, and now u1 starts to decrease. So we go to that one. So you can see here that depending on the initial conditions, one or other species is excluded. And beyond the course, this, as you know, with a saddle point, there's the manifolds stable and unstable. So here there'll be some sort of one like this, which would be the stable one. And then if you extend that further, which goes beyond the linear stability analysis, we have something that's called the separatrix. And so if you're above here, you go in that direction. If you're below here, you go in that direction. Final case, case 4, will be both less than 1. And what that means is that intersection. So alpha 1, 2 less than 1, alpha 2, 1 less than 1. u1 dot equals 0. That's 1 over alpha 1, 2, and that's now bigger than 1. And the other one is this. And this. That's supposed to be a straight line. 1 over alpha 2, 1, bigger than 1. And now we've got, again, 1, 2, 3, 4 steady states. Okay, let's look at these two. The alphas are less than 1, so the excluded ones are unstable. That's unstable, that one's unstable. This one's always unstable. If you were to do the linear stability analysis, you'd find out this one was stable. Okay, yep.
Why haven't we picked up two extra steady states where the null points intersect at 1 over alpha 2 1 and 1 over alpha 1 2?
Oh yeah, you mean sorry, that we could have them. So I'm not quite clear where that one is, sir.
So like on the axis at 1 over alpha 2 1. Why haven't we extra steady state?
Oh yeah, that's a good point to bring up. This is where the two cases where u1 dot is 0. And remember, for the steady state, u1 dot has to be 0 and u2 dot has to be 0. So it has to be different null clines for the different species, okay? So thanks for pointing that out, because I've been a bit laxed in that by just saying where the null clines intersect. What I mean is the null clines from the different species, okay? So different null clines, so thanks for pointing that out, okay? And so now what's going to happen, if we start up here, we'll go like this. Start off here, we'll go like that. If we start off here, we would cross that one horizontally, go like that. Start off here, cross that vertically, and then we can't cross that horizontally, so we can't cross it. So we go like that. Come in like that, and like that. Okay, so now we have coexistence. So we can get everything. We can get u1 to exclude u2. We can get u2 to exclude u1. We can get both of those to happen depending on initial conditions. And we can get coexistence. But notice this coexistence, I mean, what we should also do, being a bit laxed, if we started off here, we would go to there, because that's logistic growth. Logistic growth. Should draw that in for all of these. Okay, so notice if u2 didn't exist, u1 would be at 1. But now u1 is less than 1. Likewise, if u1 didn't exist, u2 would have been at 1. Now it's at less than 1. So both pay a price for there being competition. Okay. So one of the big issues about all of this concerns squirrels. So the squirrels in the red, squirrels are the indigenous population in the UK, and probably very few of you have seen red squirrels. But you've all seen grey squirrels, right? Coming along, trying to steal your sandwiches and stuff like that when you're sitting outside having a picnic. They were imported into the UK from the US around the turn of the last century. And it's still not clear why they've outcompeted the red squirrel. Okay? But the problem with them is they're a big pest, because they eat the bark of trees. They can digest the bark of trees in a way that the red squirrels can't. And therefore, they are very bad for farmers. Okay? So, there have been lots of programs set up to kill the grey squirrels. Every so often, grey squirrels come on the menu. Is another way to get rid of them. Okay? So, suppose these are the grey and these are the red. By killing the grey, what we're doing is we're lowering the population by culling them. So we're going towards the grey, and now we come in and we kill a big pile of them, have a really gigantic squirrel barbecue. And we go back to the reds. And we've won. We've got rid of the greys. If this was the case, then what would happen, just quickly draw this. Oops. And now that one is the lower one. That one, blue is the upper one. And this is the one where you go up there, because you cross the red line first. So now let's do the same thing. We're up here, come down to here. We have a giant grey squirrel barbecue. Then we come out of that to find that we're knee-deep in grey squirrels. So in other words, if this is the case, then you can't get rid of the pest by culling. You have to do this first. Or not this one, sorry. This one first. And that means you have to make alpha 2, 1 bigger than 1. Alpha 2, 1 is the ability of the grey squirrel, the ability of the red squirrel to compete with the grey squirrel. So you've got to make the red squirrel a better competitor. Right? Now you might think, oh, well, that's okay. We'll plant food that only the red squirrel eats. But the red squirrel and grey squirrel eat the same thing. Problem is the grey squirrels eat other things. Okay? Maybe we could send the red squirrels onto a fitness course. You know, get little dumbbells, have them do little exercises. That probably won't work either. Okay? So we're doomed a bit really here in this case. Okay? So let's move away from that to another example. Many, many years ago, a cancer radiologist in the Moffitt Cancer Center in Florida, Bob Gatenby, he said, why don't we view cancer as an ecology? Normal cells and tumor cells are competing with each other. And then what we do is we take the tumor out. You can never take it all out, just like you can never kill every single squirrel. There's going to be some left. Will that grow again, or will it die out? Well, again, if you're in this regime, it'll grow back again. If you're in this regime, sorry, again, I've got it wrong. If you're in this regime, it'll work. So what do we have to do? We have to make the normal cells a better competitor. How do we make the normal cells better competitors? Well, one way would be to upregulate the immune system and have both those things working together. So you can see how the abstractness of mathematics allows you to address different areas. Okay? And this idea of viewing cancer as an ecology is something that many people are working on at the moment. And in fact, we are using this model as a way to try and see, can we come up with different therapies, working together with the Moffitt Cancer Center. But of course, cancer is an extremely complex process, and to be able to find enough data to first of all validate this model and measure these parameters, that's a big challenge. It's a huge challenge. So we're still a way off yet from using these types of models to be able to advance this sort of area in disease. Okay. So then the last one is mutualism symbiosis. And this is where each species increases the growth of the other species. So there you've got logistic growth, but now you've got a plus and likewise for this one, you've got a plus. And now you see these null lines, instead of having a negative gradient, they will have a positive gradient, which means they won't go like this, they'll go like that. So I'll just sketch this. So again, you can non-dimensionalize, find the steady states, do linear stability analysis, etc., etc. I leave that as an exercise. Okay? And then what you end up getting, again, going to non-dimensional U1 and U2. And then you've got the null lines along the bottom here. And once again, you've got the 1, 0 and the 0, 1 as steady states. And again, if they're stable. But now, the other null lines are like that. So you could have something like this. That one is stable, and everybody goes like that. And you see it's bigger than it was here. Bigger than it was here. Or the intersection could be down here. In other words, like that, in which case you just keep growing, okay? But the key thing is you do better in the presence of the other species. So now we're going to leave this sort of stuff, and we're going to go on to a different area which is called enzyme kinetics. And these are not in Ruth's notes, because it wasn't on the course when she wrote her notes. But they're in J.D. Murray's book, and I put notes online. Volume 1, Chapter 6, Section 6.1 to 6.3, and I put them online, okay? And the classical reaction for this is what is called Michaelis-Menten. And that goes back to 1913. And you should know that an enzyme is a catalyst. It speeds things up. So we have S, which is some substrate, and that's got to turn into something. And the enzyme helps it to turn into that something. So the enzyme binds to it, giving you a complex. It can also unbind, and we'll put some letters here, K1 and K-1. I'll come back to them in a minute. And then when this complex breaks up, you get the product. Okay, this is not the mathematical product, this is a chemical product or biochemical product. And the enzyme comes back. So the enzyme, as you know, with catalysts, they're not created and they're not destroyed by the reaction. Okay? And so they used what's called the law of mass action. And that states the rate of a reaction is proportional to the concentration, to the product. And now here, product means mathematical product of the reactants. I should change color because it's getting not product of the concentration of the reactants. And the rate constants, so if it's proportional, then it's multiplied by a constant, and K1, K-1, K2 are the rate constants. And they're positive. And just a little bit more terminology. Here we say this is reversible. Reversible reaction. So now we've got 4 species, S, E, Se, and P. So we need to write down 4 ODEs. So we let little s be the concentration of big S. This means concentration. So S means the name of the substrate. Brackets like that means the concentration of the substrate. And so instead of writing these everywhere, we're going to just write it as little s. E equals this one. C equals this one. And P equals that one. So now ds by dt. Well, S is lost by S plus E giving you Se. The rate of that reaction is proportional to the products of the reactants. The constant of proportionality is K1, and it's a loss, so it's a minus. S can be formed by C going in the opposite direction. So that's K minus 1 times C. dE by dt. E is lost by the reaction with S. It's gained by the complex reversing. So those two are the same. But E also comes from the complex going to product and E again. So we've got a K2 times C, because the reactant there is C. What about dC by dt? dC by dt, actually we'll write it down here, dC by dt is gained by S interacting with C. And remember, this is S times C, it's not S times E, it's not SE. It's lost by going backwards. And it's lost by going to the product. And then what about the product? That's the easiest one. It's minus K2, it's plus K2 times C. Okay, so that's just using the law of mass action. And initial conditions, well, we'll start off with some substrate, we'll start off with some enzyme, and the reaction hasn't started, so there's no product and there's no C. So S of naught is S naught, E of naught is E naught, C of naught is naught, P of naught is naught. So the first thing to notice is these don't have P in them. So we could solve these and then substitute into there and get P. So P is uncoupled. And now one thing you should be worried about, enzyme is not created or destroyed, so enzyme should be constant. Here, there's nothing to say that should be 0. So what's going on here? But look, if we added these two, cancel, so that's conserved because the enzyme can either be E or it's with C. So this is conservation of enzyme. So E plus C equals a constant. Initially, it's E naught. So that's it. So now we can eliminate either E or C. And what they did was they said, okay, E is E naught minus C. So I'll just put into I'll take these two equations. So then we end up getting ds by dt equals minus k1 E naught S plus k1 S plus k minus 1 C. And I'll leave you to do the algebra. K1 E naught S minus K1 S plus K minus 1 plus K2 C, with S of naught equal S naught and C of naught equal naught. So all we've done there is we've replaced E by E naught minus C, E by E naught minus C. So what we do then is we solve these two equations for S and C. Then E will be E naught minus C, and dP by dt will be K2 times C. So what do we do? Two equations. we non-dimensionalize, find steady states, do linear stability analysis on phase planes. Okay. Now remember, I told you that non-dimensionalization is non-unique, and you can have many different non-dimensionalizations for the same thing. This non-dimensionalization is very important. It's done, and again, this is written down in the notes, you don't have to copy it down. They non-dimensionalize this in such a way that E naught over S naught came out when they did the non-dimensionalization. Okay, so again, this is done in the notes. I leave you to check. Now this looks very strange. Write this as k minus lambda. Why not just write that as something else? K1, I mean kappa or something, but you'll see later on why. And epsilon is dv by dt equals u minus u plus k times v. U of naught equals 1, v of naught equals naught, and one of the exercises in the problem sheet is for you to find the steady states, do linear stability analysis, etc., draw the phase plane. Okay? Now, why did they do this to get E naught over S naught? It's because typically E naught is very much less than S naught. So epsilon is very small. So if epsilon is very small, let's put epsilon to equal 0. We can say that's approximately 0. And that gets you v is u over u plus k. Substitute that into there, into there for v. And you can see now why we put the u plus k here, because this will be u over u plus k, and the u plus k will cancel. And we're left with du by d tau equals minus lambda u over u plus k. Okay? Because putting that in here, that becomes a 1 times u, cancels with that, and we're left with the lambda. Initial condition, u of naught equals 1. So we can solve for this analytically, and then we put in that v is u over u plus k. And then we're done. Except, what about the condition v of naught equals naught? V of naught is u of naught over u of naught plus k. V of naught is 0. And that's a contradiction. So we've done something wrong. And what we've done wrong is we've said, I'm multiplying two numbers together. One of those numbers is extremely small in size, Therefore, the product is extremely small. Okay? Right? So suppose I took epsilon to be 10 to the minus 6. And suppose I took the other number that's multiplying it to be a billion, trillion, quadrillion, billion, 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 all multiplied together. Roughly speaking, what some of these billionaires, what they're like Mark Zuckerberg's portfolio, billion, billion, billionaire. Multiply that by 10 to the minus 6. I will not get 0. I'm implicitly making the assumption that dv by dt is of order 1. But to begin with, u is 1, v is 0, so dv by dt is 1 over epsilon. If epsilon is 10 to the minus 6, dv by dt is 10 to the 6. Therefore, initially, we can't say this is 0. And that's where we've gone wrong here. So what we have to do is we have to analyze what happens near t equals 0. And that's what we will do in the next time. And the way you can think about it is this is a second order system. By putting epsilon to be 0, we've reduced it to a first order system. So think of a quadratic, epsilon x squared plus ax plus b equals 0. We know that's got 2 roots. Epsilon is small, so get rid of the epsilon x squared. We've got ax plus b equals 0, and we've got the solution, minus b over a. But that's one solution. Quadratic's got 2 solutions. Where did the other solution go? There must be another solution that is x extremely large. So the epsilon x squared, you can't put that to be 0. And that's what we'll look at tomorrow. Okay?